Got to re-screen share.
Okay.
They should do it today.
Sure.
Okay. And then you're gonna hide this again. They're all, and then you're gonna want to get rid of that.
I have no page there. Oh, here it is.
Okay.
Got him. Amazing.
All right.
Okay.
Take two.
Yay. Awesome. Thank you all for your patience. Thank you, Roshni, for the invite. Thanks for everyone. It's been great to chat with people. I keep telling everyone I visited Eugene to watch Cal men's basketball play the Ducks here about 10 years ago. We got destroyed. All the people sitting around me were so nice. They started cheering for Berkeley, like out of pity because we were getting destroyed so bad. But in spite of that, I still had an amazing time, and I've been like angling to come back to Eugene ever since then. And so I'm excited to be back. I'm going to talk about the Wright-Fisher model. It's a classic. I'll give kind of a overview first, just so we're all on the same page. And then I'm, like a methods nerd. So half is going to be methods nerd stuff in these algorithms. But I'm going to try to motivate it with some applications that span human health and also ecology, hopefully. I have a bunch of slides. I'm probably not going to get through all of them. And so just, we're not going to finish anyway. So just stop me if you have any questions. The whole point of this is to learn things. And so if things aren't making sense, definitely stop me and ask questions. Okay. So just as sort of like a broad overview, we're interested in evolution, and we want to learn things about what's happening in terms of evolution. And in order to do that, we need to be able to link these things that we care about to things that we can actually measure and observe. And the Wright-Fisher model is this sort of workhorse of population genetics that links observable quantities to evolutionary parameters. And so the way that this model works, it's very simple. You have a population of circles. These are each a haploid individual. And some of them have one allele, the blue allele, and others have the white allele. And this is an evolutionary model, so we're going to evolve this population forward through time. And the way we're going to do that is we're going to spawn a new generation. And you can think about each individual in the next generation as picking their parent uniformly at random. So I made this in Keynote and got lazy, so there's some spatial structure here. But you can imagine that this individual has an equal chance of picking any of these people as its parent. And then you inherit the allele that your parent has. So all of these individuals descended from blue parents, they happen to pick blue parents. All of these individuals, their parents have the white allele, and so they inherit the white allele. Okay, and you can keep doing this over time. You can add things like mutation. So occasionally an individual might have a mutation where they now have a different allele than their parent. So even though their parent had the white allele, this individual has the blue allele. You can imagine that over time, these things kind of bop around. There's some randomness in that people are randomly picking their parents. There's some sort of deterministic forces like mutation, and that's going to shape the frequency of this variant over time. And ultimately, we might measure individuals at present day. And so this distribution over frequencies at present is going to be related to things like the population size and the rate of mutations. Okay. And this is sort of like the most vanilla version of the Wright-Fisher model, and we can start modeling things that are like more exciting to evolutionary biologists. So for example, you might think, okay, most things are not neutral, variants are doing things, and they might impact an organism's fitness. And we can model that by doing the same process where we now have individuals with different alleles, individuals pick their parents uniformly at random, and now we're going to sort of like sprinkle on these selective events. And the idea here is if you pick a blue parent, with some probability, you're going to sort of like, you're going to get mixed, and you're going to pick a new parent. So maybe you pick this blue parent, you decided to get mixed because the blue allele is less favorable. you repick, and now maybe you've picked a parent with the white allele. And you can think about continuing this process every time. You can keep including things like mutation in here. But the idea here is now, over time, this white allele is going to end up being at higher frequencies than the blue allele, because occasionally individuals who picked a blue parent repick, and sometimes they pick a parent with a white allele instead. And so, again, this sort of generates a different distribution of our frequencies at present. And we can think about relating this distribution to things like selection. So these models tie observable quantities to these interesting evolutionary parameters. Okay, and the last sort of thing that we'll talk about in these kinds of models is migration. So you can imagine maybe this is like a mountain. I spent a long time picking which shade of brown to represent like a natural barrier. And I thought this looked very mountain-like. So this is maybe a mountain. And so now we have sort of two populations, one on each side of this mountain. And within each of them, we do the same stuff that we've been talking about for a couple slides, except you're doing it just within population. So these individuals only pick their parents within this population. These individuals only pick their parents within this population. And then you have a much lower rate of picking a parent from the other population. And so now you can imagine that if there's no migration, there's very little coupling between the frequencies in these two populations. They're evolving essentially independently. As migration gets high, it ends up becoming, you're picking your parents regardless of population. And so these frequencies are going to become quite coupled. And so there's another sort of observable quantity that we can tie to things like migration that we're interested in. Okay, and so that's sort of like the whirlwind overview of these kinds of models. And now I'll start getting into the applications. So in the first application, we're interested in sort of quantifying what genes are important in humans. So this is sort of like a cartoon of what happens in practice. I'm not a clinician, and so all of this is sort of what I think happens in the clinic, probably not 100% accurate. But sometimes you have patients that have these sort of severe, rare diseases. They present with some developmental syndrome, and you don't know what's causing it. So you don't know how to treat it. You don't know what it's going to look like in the future, and so on. And so maybe you order whole genome sequencing or whole exome sequencing. All of us have about somewhere between 40 and 100 de novo mutations. And any one of those could be the cause of this syndrome. And so how do we pick which one of those is causing the disease? Which one should we follow up on? This paper in 2019 says we don't know anything about 20% of genes. Actually, this is actually a very quantified statement or qualified statement, but we don't know a lot about what genes are doing. And even genes where we know something about their biochemistry, it's unclear, like if I disrupt a particular G protein coupled receptor, I might know something about what it's doing in cells, but I have no idea what that would look like, at the person level. So it's very hard to decide among these different variants. Okay, and in humans, we do GWAS a lot. So you might think, oh, we have GWAS, we've solved this problem. But there's a lot of issues with interpreting and linking GWAS to genes. So overall, it's very challenging to prioritize these kinds of variants. Okay, and so what we could do, and I always say, if we were mice, we could do this, but we can't. If we were mice, ethics would prevent us from experimenting on mice. But if we cared about mice, we could experiment on mice. We could knock out a gene, and this is like a classic reverse genetics screen. We knock out this gene and we see what happens. So we just learn whatever gene in the genome is. We're humans, so we can't experiment on humans. So we can't really do these kinds of experiments. It becomes very challenging to estimate what a particular gene is doing. Before I made these slides, I made this slide a year ago, and there was a Keith Haring exhibit at a museum nearby. So I was really jazzed about Keith Haring drugs. But anyway, you can think of us as living sort of a natural experiment. So one of my favorite statistics is that, in 500,000 white British individuals, we found about 50 million loss of function variants, which means that each of us have, about 100 of our genes knocked out, one copy, which is kind of crazy. That's a lot of genes, and most of us are doing, pretty well, all things considered. And so that means that we're sort of like, we are a knockout experiment. There's a bunch of us with these knockouts. But it's not really the same as the mouse situation because we didn't get to pick those knockouts. Like we're not picking which genes to knock out. If you spent any time on Twitter 5 years ago, then I apologize for subjecting you to this. But this is like a survival bias kind of problem. So the idea is like, we're not observing the effects of these knockouts. We're observing people who have survived having these knockouts. And we're also observing people who have survived sort of like on evolutionary time scales. Like not only do you have to survive having this knockout, but like if your parents had it, they also had to survive to pass it on to you. And so you can argue sort of by analogy to the survivor bias problem that we can think about these loss of function mutations as being sort of like these bullet holes in the plane. And the story here, if you don't know the story, is like if you map where planes get shot when they come back from battles, you might be tempted to think that these are sort of the important parts of the plane, but really, these are the planes that make it back from battle. And so, if you think planes are getting shot roughly equally everywhere, the planes that got shot here were fine, and the planes that got shot in the important bits were the ones that didn't make it back. And so, arguing by analogy, we might think that genes where most of us in this room, or a good number of us in this room, have loss of function variants, those genes are probably not that important because a bunch of us have it knocked out. We're doing as well as one can be in these times. Whereas if nobody in this room has a loss of function variant, then that's probably an important gene. Okay. And we are definitely not the first people to think of this. There's this big human genome sequencing consortium, the NOMAD consortium, that quantified this or tried to quantify this using this metric called LOOF. And the idea is they have some neutral model, like neutral evolutionary model, where you get some expected number of loss of function variants that you expect to see. And then you go out and you measure people and you get an observed number. And the intuition is sort of that the ratio of the observed number over the expected number should be like a measure of constraint. So if you see as many as you expect, it's probably not constrained. If you don't see any, so if O over E is very low, then it's probably a constrained gene. And they formalize this in this like somewhat complicated model where they assume that the observed number is Poisson distributed with some scaling factor times the expected number. And then they calculate a 95% confidence interval for lambda, and then take the upper limit of this confidence interval, and that's their metric. So if that sounds confusing, it is confusing, and it's confusing to us too. But people use this all the time. People have been using it. I wrote this maybe a year or two ago. At that time, it had been cited 4,000 times. Okay, so this is sort of like a confusing metric of constraint. Maybe we should think, in terms of Roy Fisher and sort of these population genetic processes and evolutionary models. And so one observation is that most loss function variants are at very low frequency. They're only appearing in heterozygotes. And so sort of a natural estimate of constraint would be, how much is my fitness reduced when I have one copy of this gene knocked out? And that's sort of like a measure of importance for that gene. And it's interpretable because in our Wright-Fisher model, it's how frequently those skull and crossbones appeared. So it's like a reduction in your expected fitness. Okay, and we call this S-hat. People call this S-hat. Some people call it HS. There's a bunch of different names for this. Okay, so now the next two slides are going to be like a little bit nerdy, so I apologize in advance. If you don't care about this, you can tune back in a couple of minutes. Okay, so I drew a bunch of circles with arrows before, and that was the Wright-Fisher model. And you can formalize that as a bunch of binomial sampling. So if X is the number of individuals with the blue allele in generation T, then if you know how many people had the blue allele in the previous generation, it turns out that it's just binomial sampling. So you're going to draw n new circles. So that's the n here. And the probability of being blue, this T, is going to be some function of the selection coefficient, the mutation rate, and then the frequency in the previous generation. Okay. And the Wright-Fisher model is amazing as like, if you do mathematics, because this is like you know, if you take undergrad probability, this is maybe like the third distribution you learn or something, maybe the second. And so this feels like this process where like we should really know what's going on here because like this is one of the first things you learn. But you do this a bunch of times in a row, and it turns out that that gets kind of complicated to think about. And so it's like at this nice intersection of being, you know, it feels like it should be very tractable, but it's also like complicated enough that there are interesting things in it. Okay, and the upshot of all this is that we can think about iterating this process a bunch of times. So we start at some initial frequency, push that forward to the present day, and then that gives us a likelihood, because this is a statistical model, so it gives us a likelihood of observing that frequency of loss of function variance given the selection coefficient. And so then we can think about sort of turning this on our head. We observe some frequency, and then now we have a likelihood of the selection coefficient. It turns out that this is a bit tricky to do sort of mathematics on. We know how to do each step sort of independently, but you have to do these steps over and over again. Okay. And so what does that look like? If I want to know what's the number of individuals with the blue allele in generation T, I'm going to do this big bit, which is probably the most intimidating looking bit, is just binomial sampling. And so I'm saying, you know, I'm drawing, this should be a J, no, this should be an I. I'm drawing eyeballs, eye blue balls out of my individuals. And then here's the probabilities of drawing the blue ball. So this is just binomial sampling. So I'm saying, what's the probability of going from J blue balls in the previous generation to I blue balls? And then multiplying that by the probability that I had J blue alleles in the previous generation. Okay. You can write this as like a big matrix. If I'm thinking about these each across different values of I here. So the probability of having 0 blue alleles, 1 blue allele to n blue alleles. And each column here is going to be like a binomial distribution. And here's my vector of probabilities in that previous generation. Okay, and you can compute this in the next generation from the previous generation by just doing this matrix vector multiply. But this is a big matrix. It has n squared entries in here, and so you're going to have to do n squared things. Okay. A bunch of people have thought about how to make this faster. So there's a bunch of work on diffusion models where basically you assume the population is very big. You're not looking at too many individuals. So you're looking at sort of common variance and then weak selection. And in this limit, sort of like the binomial aspect kind of looks like normally distributed, and then things become Brownian motion, and there's a bunch of theory to deal with that. So there's a bunch of methods based on that. And this goes back, I guess, 75 years at this point to Kimura and colleagues. There's like a familial theme here coming up, just a warning in advance. So Ryan Gutenkunst in 2009 came up with DADDY, which is like a numerical way of solving this diffusion approximation. Then there's a few other methods that came along later. This is from Simone Gravel's group. And then there's also sort of this other way of thinking about things in terms of genealogies. I'm not really going to talk about it. And Jack Ken had Mommy. So we've got Mommy and Daddy. They're based in Montreal, and apparently Maman or something is some kind of French pun also related to your mom or your dad. So these are all. But anyway, so all of this is sort of thinking about either neutral or weakly selected variance and common variance. But the variants we're looking at, we think are under very strong selection, and they're super rare. And so we didn't really want to do these diffusion approaches. Okay, and so we went back to this idea of doing these matrix vector multiplies, and we started just thinking about like what's the structure of this matrix. So each column here is a different binomial distribution, like the probability mass function of a binomial distribution. And one thing you can think about is that nearby distributions are very similar. So like, if I'm flipping a coin and it has like a 50% chance of coming up heads, and I flip it like 100 times, the distribution over number of heads is gonna be very similar to like if I flip a coin with like a 50.1% chance of coming up heads. And so it turns out that you can sort of think about grouping these columns to being like, okay, these have slightly different probabilities of coming up heads, but they're all like close enough that I'm not losing a lot by assuming that they're actually exactly the same. Okay, and then the other thing that we notice is this matrix is super sparse. So this is like a measure of how large the elements in each row column of this matrix are. So basically, like if a coin has a 50% chance of coming up heads, it's like you're never going to see 99 heads out of 100 flips. So you can just ignore, like, this is all never going to happen, because this would be like, you know, I have a very biased coin and I'm getting a bunch of heads or whatever. Okay. And so we combine those two observations. So we group these rows and then we ignore a bunch of elements that are not around. And it turns out that you can leverage these things. There's some numerical tricks you can do. When I'm multiplying this matrix by a vector, I don't actually care. I don't have to multiply this entry by this column and this entry by this column, because these two columns are the same. So I can sum up these entries first and then just multiply it by one representative column. And I can do this across like each of these color groups. So I sum up these 4 entries and then just multiply it by one representative column. Oops. And each of these columns are super sparse. And it turns out if you combine these things, you can control the amount of error in this. So like, if you want this to be accurate to like numerical precision, we can tune, you know, how many columns to group and how much sparsity to incorporate. And you end up saving a factor of n, which is a lot. So this goes from taking like hours, days to like, you know, a couple minutes. And so this is implemented in a software package called FastCTWF that you can play around with, and hopefully it's kind of user friendly. If you need to compute any Redfisher nonsense, it's nice. Okay, we implemented this. We compared this to a diffusion approximation, and it's significantly more accurate than the diffusion approximation. So here's different selection coefficients, and these are different measures of sort of accuracy. High is bad here. And so you can see that our approach is quite accurate across the whole range of selection coefficients, whereas the diffusion approximation really breaks down for strong selection. And the diffusion approximation also breaks down for large sample sizes. So if you sample essentially the whole population, even when selection is weak, the diffusion approximation is quite bad. Okay, so now this is essentially like transitioning out of the nerdy stuff and into, well, I guess other kinds of nerdy stuff, but less math and more curves, hopefully. Yeah, so these are interesting. These are likelihood curves for, you know, sort of, you can think about this as what's the likelihood of observing this frequency given this selection coefficient. And so like if I observe a non-segregating site, it gives you some weak evidence that there's strong constraint, right? Which captures this intuition that we were talking about before, that if nobody has a variant, that's some evidence that there's constraint. Whereas if we observe like a variant that's present in 10% of us, you can rule out sort of strong selection. It's unlikely to be very strong selection. And one thing that's quite interesting is that these are all sort of like only informative in one direction, essentially. Like none of these segregating sites tell you anything about what selection coefficient you have, other than it can't be too big. So like if I have a doubleton, I know it's not crazy big. If I have a 1% frequency variant, you know, I know it's not moderately big, but I don't know whether it's like tiny or kind of intermediate. Okay. So this is all just to say sort of like one variant doesn't give us a ton of information about selection, but it does tell us something. Okay. And we can quantify how much information a variant gives us at different regions of this space and for different sample sizes. And you see this interesting phenomenon where so each curve here is a different sample size. And the y-axis is a measure of how much information you get per variant. And one thing that's interesting here is that you get this plateauing effect. So like for weakly selected sites, already at a sample size of 10, you sort of have all of the information that you're ever going to have. sequencing more individuals doesn't give you more information about weakly selected sites. And that's sort of true at any selection cutoff, but the sort of sample size that you need to saturate the amount of information increases. So you can think about this sort of as like a diminishing returns argument. And we were arguing here that this is like where human sample sizes are. So we're getting like pretty close to like, we have sequenced enough data, at least for learning about selection, that like sequencing more people is not going to help, at least for learning about selection. Okay, we wrapped all this up into like this sort of empirical Bayes machinery that we call gene Bayes. I'm not going to talk about the machine learning bit, but we have this sort of machine learning back end that allows us to pool information across genes. So for any one gene, we don't have a ton of information for the reasons I was just talking about. But if we can think about ways of pooling information across genes, now we get a lot more variance and we can learn something about selection. So we have this sort of machine learning backend, and then we can relate, as I've been talking about, selection to these variant frequencies, and that's through the Wright-Fisher model. Okay, so we implemented this. Our method's called gene Bayes, and we have this. We estimate this selection coefficient. And here's a bunch of other methods for estimating how constrained a gene is. And we wanted to know, like, are our estimates of constraint any good? And this is a hard problem because, again, like, we can't knock out these genes and see what happens. So there's no sort of like ground truth here to compare against. But what we can do is look at cell line growth. So we could say, if I knock out a gene and it makes a human cell line not be able to grow, then that's probably going to be a gene that's also going to prevent a human from growing. And so we use these different metrics to try to classify whether a gene is essential to cell line growth or not. And we outperform a bunch of existing metrics, including this Luft metric I was talking about that's widely used. And the benefit is sort of increase for genes that are short. So for short genes, there's not a lot of information in one gene, and our machine learning backend that I'm not going to talk about lets us pull information. So our biggest boost is for these short genes. Okay, and while we were writing this up, Nomad went out and sequenced, well, they aggregated sequencing data from another 600,000 people. Or I guess it's 300,000. This is like a classic PopGen factor of 2 issue, but they have 800,000 haplotypes now. And so then here was on the same benchmark, here's their performance when they had 140,000 individuals, and they get a boost when they go up to 800,000, but they don't get as much of a boost as we got by pooling information across genes. And so this is really this diminishing returns thing. As we're sequencing more people, you're sort of like plateauing in the amount of information you get, and it's due to this this sort of information limit. Okay, and the last thing I'll say about constraint is it's nice because it's interpretable. We can think about this in terms of the Wright-Fisher model, and we can think about other kinds of variants that are under selection, and now compare this constraint metric to those. So for Luft, it was this sort of like upper limit of a confidence interval. And it's hard to say, like, is that similar to the amount of selection on lactase persistence or something? Or, you know, pick your favorite variant. Because those are just like, they're measured in different units. Whereas we can, you know, compare our genes to other kinds of variants. Okay. And so here's 1 minus the CDF of selection coefficients. So the way to read this is like, you know, if I draw a line down, then this is like 99.99% of genes have selection coefficients greater than this value, you know, and like 5% or whatever have selection coefficients greater than this value. Okay, and because we can interpret this x-axis here in terms of the selection coefficient, we can say that there's about a 1% of our genes, you can knock them out and they're, you know, effectively neutral in sort of as a term of art, meaning that they have a effect on fitness that's so weak that like evolution can't really see it. So there's, you know, about 1% of genes that really don't seem to matter. And these are enriched for like olfactory receptors, which is very interesting. Maybe we don't need to smell things. The next bin is sort of this like weak selection regime. And so this is sort of the strength of selection here is like knocking out a gene in this bin is like roughly comparable to the strength of selection acting on like the strongest individual GWAS variants. It's like variants that affect traits, common variants that affect traits, like the strongest selection acting on those is comparable to knocking out about 30% of genes, or knocking out one of 30% of genes. Okay. 30% of genes are sort of strongly selected, and these are sort of similar in strength to sort of like the strongest selection signals of selection that we see on individual variants across anything. So lactase persistence sort of falls in this region. It's a different story because lactase persistence is positively selected and genes are negatively selected, but you can think about this as being sort of strong effects, but maybe not super strong. And then about a third of genes have these very extreme fitness consequences. You don't really see these in like population genetic selection scans, and these are very deleterious and would correspond to having something greater than like a 2% chance of lethality. Okay, and you can plot your favorite genes here. We estimate LPA, which is a component of cholesterol, is in this weak selection regime. It's interesting. People joke that we should like put statins in the drinking water because like it seems like LDL cholesterol is not good for us, and you can like drive it as low as you want, and people are still healthy. And so like kind of makes sense that this is very weakly selected. I did tell this recently to like a membrane biologist, and he's like, cholesterol makes up 40% of our lipid membranes. Like, cholesterol is so important. Well, how dare you say everyone should have no cholesterol? So maybe that's why it's just weakly selected and not effectively neutral. BRCA1 is in this strong selection bin. So this is like a major driver of breast cancer. It's fairly common in the population for causing such a serious disease. And so there's potentially something interesting going on there. And then one of the strongest candidates is FBN1, which is like sort of essential to growth. Weak missense variants in FBN1 cause Marfan syndrome, which is like a very serious developmental disease. And so you can imagine knocking this out would be like very, very bad if sort of weakly perturbing it is having some effect. Okay, so that's the end of sort of part one. And I'm going to move on to the next application. Yeah, of course.
That plot that you showed us, Fisher's information versus S across sample sizes. Is that symmetric with respect to sign of selection coefficient? Like if you look at positively selected mutations, does it take the same shape?
That's an awesome question. I have no idea. I should have done that and I didn't. So I only looked at negative selection here. We were thinking about loss of function so much that we were like, I don't know. Also, human geneticists are super biased. Like positive selection, there's only one locus in the genome under positive selection. There. We joke about that, but we just don't think about it. It'd be very interesting to think about that. And there could have been, sweeps in the past that we just have a hard time detecting. Yeah. Any other questions so far? Yeah, okay. So the next bit I'm going to talk about is recessive selection. So all of this, like loss function variants are so rare that for the most part, we're like, again, like human geneticists, we ignore everything that's not like the most obvious thing. Like we keep reinventing things and thinking that like when we do it, it's better. And then it turns out like, people in like animal breeding have done the same thing, but way better decades ago. So one thing is like we didn't think about recessive selection at all in the past, in the previous application, for the most part because loss function variants are so rare that sometimes they show up as homozygotes, but they're usually in heterozygotes. But we sort of wanted, there's a bunch of recessive diseases. There are genes that cause recessive diseases, and so it feels like maybe recessive selection should, maybe that's something we should think about. And so we started thinking about this from the perspective of mutation selection balance. This is related to the Wright-Fisher model. Haldane sort of pioneered this about 100 years ago. And the idea is really simple. So you can think about, in this model, there's those blue lightning bolts of mutations entering the population. And at equilibrium, that influx of blue alleles has to get offset by sort of the red skull and crossbones. So we're getting new alleles coming in by mutation, And, at equilibrium, that has to get balanced by the excess death of people with those alleles. Okay, so you can write down the rate at which those things happen. So this is everyone who doesn't have a loss of function variant. They have 2 copies of their genome, and each one mutates at this rate mu, and there's maybe L positions that can cause a loss of function variant. So this is the rate at which new loss of function variants are incoming. And then that has to get balanced by this rate of excess deaths. So this is just sort of Hardy-Weinberg nonsense, but this is the proportion of individuals that are heterozygotes. They have this fitness reduction of S het, the selective constraint in heterozygotes. And then you have p squared individuals who are homozygotes, and they could have a potentially much stronger selective effect of S homozygotes. And there's a 2 here because these individuals, if they die, they lose one of their copies as an LOF, whereas if this individual dies, they lose both of their copies are an LOF, so they're losing two LOFs. Okay, if S het is bigger than 0, or quite a bit bigger than 0, you can ignore this term, and you end up with this formula for mutation selection balance. That the equilibrium frequency should be this sort of mutation rate divided by the selection coefficient. And if this S het is essentially 0, you can ignore this term. And then you end up with the frequency being about the square root of this. Okay, so these make very different predictions. These things in general are quite small. And this number is going to be very small. And so the square root of a small number is much larger than the unsquared version. So this is predicting that frequencies of homozygous, recessively selected variants should be much higher than non-recessively selected variants. Okay, so we wanted to look into this. There's a bunch of genes that we found, about 800 recessive disease genes, where when you look around, what are they doing in people? We don't see any evidence that they have effects in heterozygotes. So these are not known to cause any disease in heterozygotes, but they do cause some disease in homozygotes. And so we wanted to see, like, okay, if this model of mutation selection balance applies to any genes, it should probably be these ones. These ones look, recessive. And so this recessive mutation selection balance should explain these. Okay. And so our deal was really simple. We want to look at this frequency of loss of function variants in terms of the square root of the mutational target size and the selection coefficient. And I can imagine that the selection coefficient is your reduction in fitness. So it can't be larger than 1. Like, the worst thing that could happen to you is you die. Like, you can't have negative offspring. Maybe there are worse things than that, but not from an evolutionary perspective. And so you can plot the mutational target size. We can measure this for every gene, and then we can measure the frequency. And anything above this line of S home equaling 1, these are all totally fine. This would be consistent with different degrees of recessive selection. Whereas if it's below this line, that's worse than death. So we should not be seeing anything below this line. And then we can plot all of our recessive disease genes, and we can see they're essentially all way below this line. So either these things are somehow worse than death, or they're having effects in heterozygotes, was our. And so the sort of takeaway here is that it seems like recessive mutation selection balance does not really fit human genetic data. And we were worried about a bunch of different causes of this. So one thing that could happen is you can think about sort of inbreeding or drift as like in a small population, you happen to get more homozygotes by chance. So this is sort of like a well-known phenomenon, inbreeding depression, where you bring, you expose these recessive variants, and you're exposing them to selection. And so that can cause them to be purged sort of more efficiently than in an outbreeding population. So you can imagine maybe there's like low levels of inbreeding in humans, which we know is true. And maybe that's enough to explain, how these are below the diagonal line. Okay. I worked out a bunch of this math and I felt really good about myself. And then I was like, direct messaging somebody on Twitter, and they were like, that sounds a lot like this 1968 paper by Ned. And then I read this 1968 paper by Ney, and I was like, wow, that's exactly the same notation I used, and these results are exactly the same as what we looked at. There was no inbreeding here, but Ney worked out the small population size case. So we did extend this to the case of inbreeding. We can also use our fast CTWF machinery to compute things, but you know, we also have some pen and paper results. The disclaimer here is that our pen and paper results look like this, which is not very great. You know, it's a bad formula when you have a typo in your appendix, but you don't notice it through like multiple lines of proofreading. And then like I eventually caught that there was like a missing parenthesis somewhere. This simplifies in the large population limit to something a bit more manageable that you can think about. Phi here is the inbreeding coefficient. So if this is 0, this term becomes 0, this term becomes 0, and this term becomes mu over S home, which is exactly, you know, our square root thing. So this recovers, in the no inbreeding case, it recovers the result of Haldane, and in the inbreeding case, it sort of extends to what happens. So we did all this. This took like, you know, a few weeks of math and thinking about things. And then it really doesn't explain the data. So here is like a more realistic model incorporating finite population sizes and inbreeding. And these curves are like, before we had that diagonal line that was like the no-go zone, these would be like the new no-go zone. You know, in red, it's an completely outbreeding population, but with like a human affected population size. You can see it's like a little bit below the diagonal. There's like this like kind of curve down here. And then these are for increasing levels of inbreeding. And it would be like equivalent to everybody having their parent be, say, like first cousins would be this blue line. And if you ask how many genes fall below this line of our recessive disease genes, you need to get like into pretty strong inbreeding territory in order to explain most of these recessive disease genes. So we think that this is not really explaining what's going on. Okay. And you can measure inbreeding, at least in modern populations, and it's much lower. It's much weaker than like even for second cousins. So this is not really the explanation for this. We also don't think it's demographic results. There's this really nice paper by Anastasius and Molly Pravorsky, and where they compared the loss of function frequencies across different ancestry groups in Nomad. And they're essentially like all right along the y equals x line. So this means like the frequency of loss of function variance on average is the same in every ancestry group. So you wouldn't expect that if demographic processes were playing a large role, because different ancestry groups have experienced different demographies. Like there's an out of Africa event here, but not on the y-axis here. If there's different levels of inbreeding in different populations, then you might expect deviations here. But in the absence of that, it would be very hard to explain why these line up so much, unless it's driven mostly by selection. And selection is shared across ancestry groups. Okay, so then we thought, okay, what else could explain why these are happening, why these don't seem to be consistent with recessive selection? And our takeaway is essentially that we think that these are having sort of like subclinical effects. So they're affecting us in a bunch of ways, but they're just like, you know, affecting us a little bit. And there's enough heterozygotes that having a small effect in a lot of heterozygotes, that's what's driving the evolutionary force compared to like, you know, the fact that it causes a disease in a small number of homozygotes. And so on the x-axis here is some measure of sort of like the frequency adjusted for the mutation rate. This is like the worst axis label maybe I've ever written, but you can think about this as sort of like adjusting for sort of like what we expect selective constraint to be without having to invoke selection. And then the y-axis here is some measure of the average effect on traits. So it's the average squared effect, and it's an unbiased estimate of the average squared. Each dot here is like a bin of genes. And they're spread out by whether they're one of our recessive disease genes or whether they're not a recessive disease gene. You can see that these line up pretty well. And we've done a bunch of testing, and we're pretty sure that these are having effects on traits roughly at about the same rate as non-recessive disease genes. And so we really think that what's happening is these are having these sort of subclinical effects. And we can quantify sort of how much selection is happening in heterozygotes as compared to homozygotes. And we estimate that for most genes, almost all selection is happening in heterozygotes and not homozygotes. So it's much worse to be a homozygote. You end up with this severe Mendelian disease. But from an evolutionary perspective, it's really just acting on these small effects in heads. Okay, that's the end of the second part. Any questions? Okay. All right. So this is IE squared. And so I thought, humans, who cares about those? Let's talk about critters. Okay. And then I'm a population geneticist, so I'm not actually going to talk about critters. This is going to be very abstract, but yeah. Okay. So we wanted to understand how habitat loss affects genetic diversity. So, climate change is happening, and we're, apparently now we're building data centers, and these are all destroying species habitats. And there's been a lot of attention paid to this eliminating entire species. But recently people have been thinking about this sort of like, they've called it the silent extinction, which is like a reduction of genetic diversity caused by destroying habitats. This is a concern because you might assume, or you might think that if you reduce genetic diversity, you're sort of reducing the pool of like variation that could adapt to a changing environment. You can end up in this sort of like death spiral. And so in light of this, the UNGBF has proposed protecting genetic diversity, so setting targets. Like we want to protect 90% of global genetic diversity, which I have some issues with in terms of like how do you quantify that. But this is gaining some traction. And there have recently been some calls for like using sequencing to monitor species that are at risk. So we want to protect genetic diversity, so let's sequence critters and see if they start losing genetic diversity so we can step in to prevent them from losing more. Okay, so we wanted to use Ray Fisher modeling to think about this question. So here we're going to think about sort of like a 2D habitat. So each of these here is going to be like a patch of individuals, where within each of these patches, you can think about this as being one of the populations we were talking about before. And then across these patches, you can think about the patches as being this brown line. That's why these boxes are squiggly brown lines, because it's the same squiggly brown line. So, individuals here are mostly having offspring that end up here, but occasionally they're having offspring that end up in adjacent boxes. Okay, and then we're going to model habitat loss by just reading some of these boxes. That's right. And then we're going to measure genetic diversity in these remaining boxes. Okay. And genetic diversity here is kind of a loaded term in population genetics. So here I'm explicitly referring to pi, or heterozygosity. And so it's going to be 2 times the frequency times 1 minus the frequency. And we can sort of average this across picking individuals at random from across the genes. Okay. And so now here, one thing to note is that this only depends on the frequency, and this pi, this thing is big when the frequency is close to half, and it's small when the frequency is close to 0 or 1. And so we're in this sort of like common variant regime. This is dominated by common variance. So this is where the diffusion is a good approximation. So in the first part, I said, oh no, we never want to use the diffusion. And now here I'm going to say, let's use it. So here's the diffusion. You can write down, this is a stochastic differential equation. So this is relating the change in allele frequency to these forces we talked about, mutation, migration, and then this captures genetic drift. And if you haven't seen a stochastic differential equation before, this first bit is exactly just a normal differential equation, like an ODE you see before. And then you can think about this bit as being like, you're going to add like a little bit of random Gaussian noise at each step. And this ends up being like the differential of a Brownian motion. But you can think about this as sort of like a noisy differential equation. There's this really nice result called Dinkin's formula, where essentially you can just take like the expectation, the average of both sides of this equation, and just forget that like I have like a differential going on here. And it turns out that you're like allowed to do this. There's no reason up front why this should work, but you're allowed to do this. So we did this, and that lets us turn this thing that's like this noisy ordinary differential equation into a system on these expected values, these moments. And this is now like just a completely ordinary differential equation. So we've turned this thing that's like noisy, and like this is like, you know, I did take a grad school level class to understand what this was saying. Whereas like an ODE class, I had to take like an upper division undergrad class. So we've turned this from like a really annoying math, but this is not so bad. Okay. Yes.
Oh, just gonna let you know, we're about 5 minutes to the hour. I know you said a lot, you had a lot of slides, so.
Yeah, okay.
Just-
I should be done almost on time. I'll end on time.
Yeah.
Thank you. Yes, so we have this system of ordinary, we have this system of ordinary differential equations. You can numerically solve this, which we have implemented in this package called WF moments, if you want to look into this. And we kind of use like, this system ends up being sparse, so there's some tricks you can do to make this faster. And the upshot of all this is that a bunch of things that we care about, including genetic diversity, things like FST, these are just functions of the first two moments of the frequencies. So we can use this machinery to compute the dynamics and equilibria of these frequencies. Okay, so now onto habitat loss. So we did spatial simulations, and then we used the theory that I talked about, where we have these critters evolving on a landscape. We remove half the landscape, and then we allow them to keep evolving forward in time. And one thing that's very interesting that we notice right away is that genetic diversity actually does not go down very quickly. So this is, you're destroying half of the habitat, and there's essentially no short-term change in genetic diversity. And it takes like thousands of generations before you start feeling like the full effects of genetic diversity. So if you're talking about like a tree that lives for 30 years, like you're talking about like, you know, 30,000 years or something, like a very long time before you feel the effects. We found that the geometry of the habitat plays a role. It's a bit interesting. I'm going to skip all of this. And sort of whether the habitat is fragmented versus whether you sort of are deleting a continuous portion plays an interesting role. But I'll just end with some takeaways here, which is that what was surprising to us is that genetic diversity really doesn't change immediately following habitat loss, but it does keep decreasing. Even we just destroy habitat and then let the population evolve, and genetic diversity keeps going down. And so our takeaway is that in practice, like genetic monitoring is probably useful, but by the time you see something in the genetic data, it's already way too late. Like you've already destroyed way too much habitat, and it's been on this like sort of downward trend for a long time. Okay, I want to save time to thank everybody. This was a bunch of different projects. The constraint on ELIS was led by this really wonderful grad student, Tony Sung. Selection on recessive genes was led by John Flood and another amazing grad student. And genetic diversity after habitat loss was with Christie and Clemens. These two were both in Jonathan Pritchard's lab, and this was with Moy at Berkeley. Okay, and I think I have 1 minute for questions if anyone has questions.
Once you're on this like standard, there's habitat restoration. Is it already too late?
Yes, it's the same story kind of in reverse. We have some in the supplement of our paper, where like you allow it, you know, destroy habitat, it goes forward for a while, and then you restore. And it's essentially the same story, but in reverse. It takes a while to come back again. So everything is slow for genetics.
I had a question back earlier when you were talking about calculating strength of selection on human genes and, for example, I think 1% is very effective. It's for sure accuracy. So like, do you see any other general trends when you look at that, like that large gene families that have lower selection if they're redundancy, for example, or other? I
Yeah,
imagine there's all kinds of interesting questions.
yeah. So we used a bunch of features. Some of them are like kind of cheating. So like one thing that we find matters a lot is like how constrained missense variants are. So we're looking at loss of function variants. So genes where the missense variants also look constrained are constrained. So I don't think that's like a very biologically interesting answer, but there are some interesting ones. So expression explains a lot, and like protein embeddings and gene structure. So I'll show what these look like. Sorry for the sideways plot, but this is like a measure of how important a set of features are. And so this says that expression patterns in brain, digestion, and development are all quite important for predicting constraint. So we think where genes are expressed is very important. And then the other bit was we generally found like the more like complicated a gene is, the more constrained it is. So yeah, so like genes that are like highly connected in protein-protein interactions or co-expression networks, if they have a bunch of enhancers or promoters, or if they have like long UTRs or a bunch of different isoforms, these are all predicted to have much stronger constraint. In terms of gene sets, I don't have a slide. All genes are kind of all over the place. So we didn't see any like clear patterns in terms of like Go or like olfactory receptors are definitely enriched for weaker selection. Mendelian disease genes are way enriched for strong selection, but everything else was kind of right. All right, thank you all so much.
There is training lunch and 3 pieces today. Pizza, and yeah. Pizza is on the menu.